Well, good morning, ladies and gentlemen. I've been invited to talk to you on some studies which we've been doing over the last 2 years to, as George told you, to incorporate mesozeaxanthin into eggs. Now, the people that have done this work, of course, have been the people in Waterford: Katie Mayer, Rachel Moran, John Nolan, Stephen Beattie. They were the people at the workface who've done most of the work, and Katie was doing a PhD project out of this. Our role, myself and Alan Howard, was advice and guidance as things went along. Now, the background, can you not hear? How do you fix this one? Sorry about this. Okay. Can you hear me better now? Good. Well, most of you know the objectives which, or the background to this sort of work. The xanthophylls are important in the macula. Supplements containing these xanthophylls will enhance visual function and are believed to protect against age-related macular degeneration. There's very little MZ, as George has just said, in the diet, but the chicken egg is known to be a particularly highly bioavailable source of lutein and zeaxanthin, but contains no MZ. So our objectives in this were to produce an MZ-fortified chicken egg, and then to feed this to volunteers. And I should just say at this point that there are 2 posters which you should consult if you want to have more details than I will explain this morning on each aspect of these studies. So these are the chickens we used. They were called gold line hens. At 40 weeks, that's their optimal, when they become of age, producing an optimal number of eggs. We fed xanthophylls at 140 milligrams per kilogram. That's the free xanthophyll. We had 8 groups in the first experiment, 5 hens per group. New feed mixes were prepared every 2 weeks. They were vacuum packed in 1 kilogram amounts, and stored at -20 to ensure stability of the xanthophyll in the feed. We used a complete grain feed because it was easier to mix the xanthophylls with the feed than if we'd used pelleted feed. Now, these were the supplements which we investigated. There's lutein, zeaxanthin, and mesozeaxanthin, and the diacetates of each of these. And at the bottom, we had 2 mixtures of MZ diacetate and lutein, a 1-to-1 mixture and a 3-to-1, where we had 3 parts of the MZ to 1 of lutein. The reason for the last 2 mixtures was because we wanted to try and achieve an egg which had roughly equal amounts of lutein and MZ. The extraction of the yolks that we did, we collected 4 eggs each week. Yolk was suspended in phosphate buffered saline. 0.1 of this was and stored at -80 until we were ready to use it. 0.1 of the suspension was then hydrolyzed using aqueous KOH. Internal standard was added, antioxidant was added, BHT, and it was incubated for 45 minutes at 45 degrees. Again, that was stored until we were ready to analyze. And the procedure for measurement was a 2-stage procedure, as was described last year, and has been modified slightly, but it's still essentially the same procedure, but I'll show you brief details. Once the sample have been prepared, we extracted twice. We extracted twice with methyl tertiary butyl ether hexane mixture, dried, stored at -80, reconstituted in the mobile phase that we wanted to use. Now, the first assay was done on a reverse phase column using a stepped gradient. The objective of this was to have a nice long time to separate lutein and zeaxanthin, and then we changed the gradient so that everything else came off very rapidly, and we could then continue with the next sample. Now, we collected the lutein and the zeaxanthin, the mixed zeaxanthin isomers from this system. Again, stored at -80 prior to assay when we collected the mixed zeaxanthin fraction. Most recently, Katie had developed or used an automatic fraction collector which collected and dried automatically, which was a big improvement on old systems. And the separation of MZ and Z was done using a normal phase column with a continuous gradient. It's a chiral column using low temperatures to improve the separation. We took the mixed fraction, we dissolved it in 100 microliters of hexane IPA, which was the mobile phase, injected 50 microliters. And from this, we obtained the proportions of the two isomers in a very good separation. Now, with the proportions of the two isomers from this assay and the known quantity from the first assay, we could quantify the amounts of lutein, zeaxanthin, and mesozeaxanthin. And these were the results from this experiment with 8 different feeds. There's quite a variation. These are the total yolk concentration of xanthophyll. So you can see that free zeaxanthin produced the lowest response, whereas the diacetate produced the highest response. The two mixtures that we were particularly interested in, there was virtually indistinguishable. There was certainly no statistical difference. And we chose to use, on the basis of this study, the one-to-one mix of MZ and MZL, the lutein MZ diacetates. These are just bringing out some of the points. You should note that esters gave higher responses for the particular xanthophyll in all cases. And we selected, as I said, the one-to-one mixture for the human feeding study. The actual figures at 6 weeks, I should have said, you notice that we achieved a plateau concentration at around about 3 weeks. And the plateau concentration of yolk xanthophylls, was lutein 34%, zeaxanthin 22%, and MZ 44%. The total xanthophylls for treatment, 1.67 milligrams, that is 9.5 milligrams per 100 grams. And that had a proportional composition of 7 to 5 to 10 by weight. So, a human study. Single-blind study, recruitment and feeding to control and treatment subjects on 2 sites. This was necessary because it was obvious that if we'd fed them in the same room, those receiving the controlled diet would have known very easily which diet they were receiving. The difference in the egg color was very large. They received a daily breakfast, 5 days per week for 8 weeks. There were 50 subjects altogether. Cholesterol we looked at particularly was less than 6.5 millimole in all cases. There has been concern in the past that eggs will increase cholesterol, and we only had to drop 1 person who went above 6.5 millimole per liter. The groups contained 25 on the controls, 25 on the treatment. And all subjects received 2 egg equivalent of scrambled egg breakfast. The objectives, obviously we were measuring serum and yolk, carotenoid, xanthophylls. We measured macular pigment optical density using 2 techniques, the heterochromatic flicker photometer and used the spectralis. For visual performance, we looked at visual acuity and contrast sensitivity. Now, the preparation of the eggs, as you can see, it had 60 control hens and 60 treatment hens. The treatment hens were fed 70 milligrams of lutein and MZ diaster salts per kilogram of diet. The eggs we decided to discard for the first 4 weeks, although we believe that 3 weeks was all that was necessary. We discarded for 4 weeks to ensure that they were on plateau. The total number of eggs collected was 6,120, so you can see this was quite a large study logistically, even though we only had 50 subjects. And 4 randomly selected eggs from both groups were analyzed for xanthophylls each week. This shows the total yolk composition in the treatment eggs and in the control eggs. The 6-week point, which we achieved with the first experiment, was there. And you can see we have roughly 1.5 milligrams per egg. We had 1.67 in the previous study. It's interesting, and I point this out at this stage, that it goes up after the 6 weeks and then comes back down again. And this is mainly due to changes in MZ. The lutein was more or less constant throughout. That may explain a slide which comes later. This was the data from those eggs. I took the average content of xanthophylls for the entire period in order to calculate these figures. So for the control yolk, we have lutein, what are the units? Milligrams, micrograms, sorry. Treatment yolk, those. If we take a mean intake of 1.4 eggs per day, because we've got 7 days, we only fed for 5 days, so we used 1.4 egg equivalent. For the controls, those work out at that intake of lutein and zeaxanthin, and treatment 771 and 732. This was the breakfast. I'm afraid the slide looks a little bit fuzzy from here, but I hope you can see it more clearly. The egg was an intense yellow, as you can, I think you can see in that slide. We received eggs plus toast on Mondays, Wednesdays, and Fridays. Croissants and muffins for a change were used on the other 2 days. Now, both eggs were prepared at the Institute kitchens. The treatment breakfast was transported to a separate site and accompanied by tea, coffee, and orange juice. Absentees given 2 eggs to eat at home, and I've already mentioned this particular point. Right, the next slide shows the baseline results. Numbers 25 in each group. Now, it was unfortunate that we had a very big difference in the sex distribution in the 2 groups. And this was because on the treatment site, although there were a lot of women in John's group who were based on that site, they couldn't be members of the study, obviously, for obvious reasons. And the only other group up there was principally men. And so we had very few men on the treatment diet. Smoking, very similar proportions, 20% and 12, not significant. Mean age, slightly younger in the treatment group. Macular pigment, optical density, there were no differences with either machine, either technique. Visual acuity, no difference. Serum lutein, no difference. Zeaxanthin, no difference. So what do we get for the results? Well, I separated by sex because I believe that the sex does influence carotenoid absorption, carotenoid responses. And also we had this big difference in the distribution of the sexes in the 2 groups that it was essential we looked at sex separately. And just as you can see in the far side, where we're looking at women, women responded very highly to the treatment group. There's a highly significant difference in those that received the treatment eggs to the 14 who received the control eggs. In the men, we had no difference between the two groups. There was a significant difference between the women and the men who received treatment groups, only just achieved significant difference. But you're dealing with 3 subjects here, so it's not surprising that we did get a difference, a significant difference. That was lutein, sorry, I should have mentioned that on the previous slide. Here we have the MZ response. And I point out that at 4 weeks, we had higher concentrations in the serum than we had at 8 weeks. And you'll remember that distribution of MZ concentrations in the yolks of the eggs. I don't know whether that explains that fall off that we appear to get there. The women have significantly greater response than the men, as you can see. And though, of course, there was no MZ in the control group. Next slide. Macular pigment optical density. Unfortunately, there was no significant difference here between the groups, though we did see evidence that in the enriched group, the macular pigment optical density did appear to start rising, but there are only 3 subjects. We saw a similar picture when we looked at 0.5 and we looked at 1, and we saw a similar picture with the spectralis results. And had we had more women, we may well have got a significant increase, but we didn't, so we can't say that there was an increase. And there were no significant differences between the control for best corrected visual acuity or contrast sensitivity, so I'm not showing any more of those results. What I do want to spend just a little bit of time on is the responses we got in comparison with pharmaceutical preparations. Although we showed no clinical benefits from the treatment eggs, the serum response was much more efficient than we'd found previously with dietary supplements. The table will show you the serum lutein and MZ concentrations, the next one, after 8 weeks of egg intake, compared with MacuShield and lutein plus. These are 2 preparations which we currently use and we have used in the past, which contain MZ. So what we have on this busy slide is that these are the eggs, women and men. The lutein content in 1.4 eggs is 0.771 milligrams per day. The MZ content was 7.32 milligrams per day. The concentration in the serum in the women and in the men. And then what we've done is we've expressed those concentrations per milligram of intake of carotenoid. So you go up, because there's less than a milligram here, to 412 and 304. And likewise with MZ, we go up from 1.31, 078 in the men, to 179 and 106. Now in the other 2 columns, you see MacuShield at 8 weeks, lutein plus after 3 weeks. The content of MZ in lutein plus was 8 milligrams a day, and these supplements were actually measured. And this was work I did in Coleraine before the work was taken on in Waterford. These were done at 3 different times. So you'll say, well, there are probably error differences in the methods. But in fact, as I say, I was responsible for the measurements here. I advised and assisted the group setting up the methods in Waterford. And so Katie was using the same principles in the methods which were done in Waterford. So I think that the results are comparable. And although Katie had considerably improved the methodology for the egg extraction, it's still the same principle. So I think the results are comparable. And if we say the results are comparable, then the relative impacts of the MZ per milligram of dose, if the response for MacuShield is taken at 1, then the lutein and lutein plus is between 5 to 7 more effective, and MacuShield eggs are 18 to 30% more effective. So there's considerable more effectiveness in carotenoids presented as eggs than there is for pharmaceutical preparations. Now, I spoke to Carlos about these results a couple of days or yesterday, and because they prepared the supplements in both cases. The lutein plus apparently was made up in soybean oil. The MacuShield is made up in sunflower oil. I don't know if that is the reason that we got better responses from lutein plus than we get from MacuShield. These are quite consistent results. We get much lower responses from MacuShield than we got from these results for lutein plus. The reason for this we don't know. Different oils may make a difference in the absorption. Different size of oil particles, the size of the particles may make a difference in the absorption of these things. The lipophilic mixture that's present in eggs obviously makes quite a big difference to the absorption and concentration that you achieve. So summarizing the results on that last slide, we have a 5-fold efficiency factor for eggs by comparison to lutein plus, and a 20-fold efficiency factor compared to MacuShield. So are we making too much out of this? After all, there were only 3 women in that study, but in fact there's quite a lot of evidence in the literature that there are differences between the sexes in the carotenoid area. Higher concentrations of beta-carotene, alpha, and beta-cryptoxanthin in women are reported by a large number of authors. As I said, we previously reported the difference. Women responded better to lutein and MZ in lutein plus. Berkmans, who's talking later this morning, published a study in AJCN in 2002 where they looked at 376 adults. They reported all serum carotenoids were higher in women. Lutein was higher in adipose tissue and also higher in women. Macular pigment optical density was 13% higher in women. So there is evidence that women respond better to carotenoids and xanthophylls than men. The reasons for this, well, there are potentially many, I suppose, but we still don't know the real explanation. Well, we don't know all of these. So in conclusion, it is possible that women responded better to the egg xanthophylls than men. There were no significant differences in visual measurements between the control and treatment group. But as I said, had we had more women, we might have seen a difference in macular pigment optical density. The lack of conclusive changes in visual functions may have been due to the relative young age of the volunteers. There were no cases of AMD. The good health status. These were academics working on the site, or potentially academics in the institute control side of the trial. Or was the trial too short? This was an 8-week study which involved a lot of work. But we don't know the answers. We acknowledge the thanks for the Howard Foundation that funded the study, and the authors are very grateful to Industria Organica, Mexico, who supplied the supplements. The references are shown on the posters if you want to look at them. Thank you very much.
Thank you so much, David. Questions? Toss.
Hello, thank you for the talk. I want to debate you on the, here we are, on the relative concentrations. I don't think you can compare a 10 milligram supplement with a 1 milligram egg and then say that it's more effective. That's I think what you should have done is a 1 milligram supplement and then do this relative comparison, because it might well be that if you have done so, then I could come up with your conclusions, but comparing 10 milligrams and then dividing by 10, I don't think that's a way to state that it's really more effective.
But had we used 1 milligram, we would have seen no evidence probably in the blood.
If you would have you should have used 1 milligram supplement to do the comparison as you did.
I agree, yeah, but
so then,
whether we would have had the results if we'd done that, we'd have had much greater variability between subjects in response.
Just to make an important point, this was the exit study. It wasn't a comparison of supplements to eggs, and David wanted to make the point of the differences that he sees. But I agree with Toss. The experiment wasn't conducted to compare supplements to eggs, and I don't think it's comparable. I agree with you.
I agree with that. It wasn't done to that, but...
Well, you made a big point of it at the end, so that's why I commented.
Yeah, sure.
Are we sure that all these lutein in the plasma are measured in fasted conditions? Are these steady-state lutein level, or is this postprandial lutein level?
Well, the Macular study was an 8-week study, and we in fact went on much longer.
No, they are not.
Is that what you're asking?
But this is important because chylomicron goes up and down very quickly in the plasma. So
These were plasma concentrations.
yeah, but this is total. This is chylomicron and other lipoproteins.
In the plasma, we extracted the plasma
Yeah.
and took it up in organic solvent.
Yeah, but at steady state, lutein and zeaxanthin are carried mainly by HDL,
Yes.
which has a very long half-life, whereas chylomicron, which is postprandial, goes up and down very quickly, a couple of hours. So if we don't take into account the pharmacodynamics and kinetic of lutein and zeaxanthin in different lipoprotein, it's very difficult to compare.
In other words, did we use fasted or
Yeah.
fed samples?
Yeah.
I don't think we took...
On the day of study visit for serum, they were fasting for that.
Yeah.
Well, my point on fasting is I understand but I don't think when because I've done experiments comparing fasting using versus non-fasting blood samples, I see no difference. And with respect to clinical trials, therefore, and the logistics around it, I think, particularly when you extend that into elderly populations, I typically do not ask subjects to fast every morning.
Yeah.
But for the exit study, the subjects were had fasted the morning of the investigation.
Yeah.
So.
But you don't expect lutein and zeaxanthin to be distributed similarly in different lipoprotein in fasting and postprandial conditions.
My question is, how come you use fried eggs rather than poach the eggs? Because longer you cook an egg, you change the content of the fat. It may be the same with the carotenoids, and maybe won't be absorbed as well. The least amount that you cook the egg may affect it rather than frying it, and that may be why the macular pigment didn't change.
Well,
Although maybe you started with too high of macular pigment rather than low
Yeah,
macular pigment in the subject. I was wondering if you thought about that. People actually, there's some people in the States that actually recommend eating eggs raw if they're natural. Obviously, you can't do it to get the most benefit. And so I was just wondering why you decided to fry it rather than poach it.
I think it was convenience to a large extent. Preparing scrambled eggs and using a dispenser which was roughly equivalent to 2 eggs was very convenient when you're dealing with a lot of breakfasts.
Because the more you fry it,
John
the
wants to
more
make
you
a
change
comment.
the fat content, and may be the same with the carotenoids.
We actually scrambled, and we did an experiment. David didn't go into it before. On the decision to how we gave the eggs was actually informed from an experiment as well, and we compared scrambled to fried to poached, and scrambled, A, for convenience, but B, for the concentrations that we were able to get in there, was the best decision to make for that trial. The failure of the trial, and it was my fault, the failure of the trial was that we simply did not follow the subjects long enough. And you may ask, well, why? The expense and the logistics of continuing the trial and paying chefs to feed in a very controlled environment, I believed wrongly, based on the previous literature, when we look at Adam Wenzel's work and other work from the United States, that suggested that normal eggs in a period of 8 to 10 weeks have a significant change in macular pigment. The extension from that was that if we had these fortified carotenoid eggs, surely within 8 weeks we would get that response. And for me, that really questions some of the previous egg work that has been published in terms of its ability to change retinal macular carotenoids. I just, based on our experiment, I now question that previous data.
Excuse me, Dave. I'm sorry. My first comment was along with Tos, is that there is a kinetics, and as the concentration increases, the efficiency of absorption will decrease. I think that it's probably not a real fair comparison there. The other thing is related to sex differences with carotenoids. That's seen pretty regularly. I know the work that we did with Japanese quail, I mean, different animal obviously, but there are huge sex differences among different species related to carotenoid metabolism, and it is interesting to know what the sources are.
Thank you.
Last question.
Just a question about, we know that plants are obviously showing different production of xanthophylls at different times of day and light and temperature. There's no reason to think that animals don't do the same thing, that they might uptake it differently at different times of life, different times of year, even different times of lunar cycle. I'm just wondering how much of that could play a role in some of the results you're seeing in the comparisons between studies, but also between comparisons in sexes, where you have women that might be still in childbearing years that are therefore going to uptake xanthophylls more quickly in order to incorporate that into their eggs. But I'm just wondering whether that could play into some of these experiments.
You're asking how carotenoids are taken up at different stages of life?
Yeah, different stages of life, also different time periods. So lunar cycle, different times of year. So naturally our diets would change at different times of year. So you're going to take in different proportions of meats and vegetables. So we might expect an underlying change, seasonal change in our ability to take up those xanthophylls.
Seasonal changes we don't get very much these days. I mean, go back 40 years and you would see big seasonal differences from the consumption of different fruits and vegetables. Through life, well, lutein is present in breast milk, we found that, so it's obviously there for the child very early on, even though the child may not be consuming lutein, apart from any milk that he consumes. In the younger years, carotenoid beta-carotene tends to go up in the serum as time goes by, although people tend to consume less vegetables as they get older and they have problems with their teeth and chewing them. And as George made at the point, it's very difficult chewing green vegetables, and certainly the Brits don't like very much in the vegetable line. I can't really answer the question any more better than that.
Thank you.